Thank you for joining me today as I share a little bit of the work that my lab group is doing here in the biology department at East Carolina University. So my lab studies complex adaptive systems. We're interested in how cells transduce energy to perform action, how cells compute their physiological states from information in their local microenvironment, and how energy and information scale from the molecular level to the cellular level to the cell collective level to inform the organizing principles of complex tissues. Now, the model system that we like to use is the process of fertilization, and we particularly like to focus currently on male gametes. And the reason is because they are a fantastic model of understanding how cells with very minimal cellular functions can still perform complex tasks. So, for example, here's a sea urchin sperm that's responding with chemotaxis to a photoactivatable chemoattractant. These are C. elegans sperm that are responding to membrane depolarization by sprouting these little potopod feet and crawling around and searching their local environment. And though not quite a sperm, this is a pollen tube that is following a ovum that's been attached to a robotic arm. And what's important about all these processes is that these cells are clearly doing complex things, but they're doing it without the aid of changing their transcriptional programs. And they're also doing it in completely different ways. So, for example, the motor protein system that underlies the C. elegans sperm motility is very different than the system that underlies flagellated sperm motility, like what you see with the sea urchin. Now, my lab specifically studies mammalian sperm. And if you were a mammalian sperm and you wanted to be successful, like this mouse sperm here, you would need to move in such a way that you increase probability of contact with the egg. You would need to stay alive outside the environment that you developed in long enough to find the egg. And you would also need to be able to recognize that what you bumped into was an egg and not something else. All these processes are mediated by complex biochemical changes, and together we call those capacitation. So they gain the capacity for fertilization over time. And importantly, a typical mammalian ejaculate can contain hundreds of millions of cells, but only about 10 to 100 or so will even make it into the vicinity of the egg. So there's a very strong selection process acting on sperm during fertilization. And that selection matters. So it's imposed by the nature of the microenvironment. So, for example, here's a mouse oviduct. In humans, we call that a fallopian tube. And sperm have to navigate their way through this incredibly complex environment that contains epithelial folds that create spatial limitations, that also have very different gene expression profiles that change the luminal chemistry in different regions of the oviduct. So in order for the sperm to reach the egg, they have to transition from this uterotubal junction all the way through this kind of maze-like structure, finding the egg down here in the ampulla. And here's a histology slide to kind of show approximately what that looks like. And to give you a sense of scale, I'm now adding a little red line over here to show approximately what the size of a mouse sperm in that reproductive tract would be. Now, organisms, as they get bigger, have bigger oviducts. So this is a human fallopian tube, transverse section. But human sperm are actually smaller than mouse sperm. So the sperm don't get bigger as the reproductive tracts get bigger. And this, here's a human sperm that would be approximately 65 microns. So as you can see, it's a small cell searching in a very, very complex spatial environment. And there are two general effects on sperm fitness of this selective process. So first, there's stabilizing selection. So features that make sperm more likely to be successful are stabilized through inheritance, and that takes the form of apparent competition. And then there's purifying or negative selection. So features that cause the sperm to be less likely to fertilize end up not being inherited or represented in future generations. And sperm selection really does matter. There's great evidence for this. So it influences development and the likelihood of fertilization. So some good evidence from human clinical data is that pre-selection of sperm, so applying things like differential centrifugation gradients or hyaluronic acid binding selection prior to intracytoplasmic sperm injection, which is where one single sperm is sort of sucked up into a glass micropipette and then injected directly into an egg. Pre-selection of the sperm can influence the fertilizing efficiency and live birth rates by about 50%. So the sperm that's chosen really matters. There's also additional evidence in cattle breeding where they can do in vitro fertilization and kind of natural fertilization using the same mate pairs. What they find is that in vitro fertilization can cause allelic ratio distortion in the offspring. There are natural cases of allelic ratio distortion, such as the T haplotype in mice. And in fish, since they're external fertilizers, there's some really great work where experimental selection is applied to the sperm, and then the developmental trajectories of the resulting embryos are followed. And ultimately, applying selection to the sperm can cause things like slower development. So for many reasons, the process of capacitation, the way that sperm mature prior to fertilization that enables successful fertilization and may influence developmental consequences, has been of interest for quite some time. And most of the approach to understanding its regulation has been focused at the molecular scale. So through lots of transgenic mouse experiments, knocking out different ion channels and second messenger systems, such as the soluble adenylyl cyclase system, has ultimately developed this really nice molecular pathway that's fairly conserved among mammals. However, what we're mostly focused on is trying to understand if that mechanism is applied across many cells that have individual variation, then how do we make better predictions about the overarching process of fertilization? So being able to identify specific mechanisms that underlie biological processes sort of invokes a very particular kind of philosophy about how living systems are organized. For example, if we imagine a biological cell as a clock, then that clock has a finite number of pieces. If we were to dissect the clock and examine the pieces and catalog them, and then put the clock back together, knowing their initial states, then we should be able to predict the future behavior of that system. However, biological systems really don't work that way. It turns out they're more complex, meaning that they have properties that are much more difficult to predict, even if you know the initial states of the components that make them up. So one of my favorite teaching examples for this is a murmuration of starling birds. And we know what the components that make up this process are. They're birds. We know what the initial states of the birds were. 10 minutes ago, the birds were all hanging out in that field eating bugs. But now they're doing this complicated and very clear patterned motion that is really, really difficult to predict from just the initial states and the components alone. So our interest in the lab is trying to understand how do more complex emergent behaviors arise in large populations of cells when mechanistic things like cell signaling processes are implemented with random inputs from the environment. And the way that we do that is by using slightly different forms of models. So the molecular pathway that I showed you previously is a type of model. It's a model of cause and effect molecular interactions that are implemented below the scale of the individual cell. Ways that we can do this for larger cell collectives or more emergent patterns might be to use things like agent-based models, which have been around for quite some time now. So this is a GIF of Conway's famous Game of Life. And what's happening here is the individual pixels in the image have a simple set of rules that changes their color based off of the current colors of their nearest neighbors. And importantly, these patterns that you're seeing are not encoded anywhere in that rule set. Instead, they emerge from the interactions of individual cells with each other over time when there are random inputs. And that concept of agent-based models has sort of burgeoned into a bigger field known as artificial life, which is doing some really cool things like trying to understand how more complex tissues can emerge from cell-to-cell interactions, such as the case of this kind of apparent vascular network. So to build a model like this, the process is actually pretty straightforward. First, you define the environment, then you define mechanistic agent rules, and then you simulate with random inputs. So rather than trying to implement these kinds of models for the in vivo environment, which would involve modeling the reproductive tract, which we felt was too complicated and not experimentally tractable, we instead chose to use in vitro sperm motility analysis, which is a common clinical measure that's often used for understanding sperm physiology. So this represents sperm would be placed on a glass slide and then sort of sandwiched between a cover slip with a spacer. And then we can observe the sperm motility patterns by using differential interference contrast imaging. And then the movement patterns of the sperm can be quantified by tracking the motion of their nuclei, which show up in the microscopy video. So if this was the nucleus over time, the sperm would be sort of swimming along this trajectory. And we can use movement parameters such as average path velocity, straight line path velocity, curvilinear velocity, amplitude of the lateral head displacement over the average path to describe the overall movement characteristics of the cells. And in simulation, then what we can do is create a particle that simulates the sperm nucleus. And then over time, we can have it choose an angle to move in and a step length using random inputs from the environment and intrinsic properties of the sperm. And then by repeating this over time, then we get movement patterns that can look quite a bit like real sperm motility. So to make it behave like real sperm, we needed to first gather data from real sperm. So here are some examples of temporally coded images that show different kind of categorical movement patterns. So here's a progressive linear pattern. So the sperm is moving along this trajectory. This is a 40x image of individual mouse sperm. This is an intermediate pattern whereby they move more laterally and a little bit more slowly. Here's a hyperactive pattern whereby they move asymmetrically or in circles like this. And then there are slow and weak patterns that have ultimately relatively low motility. So to make our agent-based models behave like real sperm, we looked at distributions of real sperm motility parameters like curvilinear velocity, straight line velocity, and average path velocity. And then we fit our models so that the sperm that we created, the digital sperm, would have movement parameters that were approximately the same as or centered on the distributions of the real sperm, which gives us a pretty nice result. So here's a 10x phase contrast image of human sperm. And here's a model simulation showing our agent-based sperm that are moving, and I hope you can appreciate, pretty much the same as this real sample of sperm. And importantly, there's a lot going on in this model. So there's a complex distribution of different sperm motility types that are mimicking the kinds of motility types that we have in the real sperm videos. So using these models, then we can sort of probe specific questions about how sperm search space and how their behaviors collectively scale from single sperm to many sperm. So in these simulations, we're just doing a short simulation where the sperm are sort of searching an enclosed space, and we have each motility type represented here. So we have progressive, linear, intermediate, hyperactive, weak, slow, and then we have a mixed population that has equal proportions of each type. And in each one of these simulations, the total number of sperm is the same. And we use the root mean square displacement as a measure of the average amount of movement that each sperm made within each simulation. And importantly, linear, progressive, and intermediate motility types moved the most, which may be not too surprising. But interestingly, the mixed population on average moved in kind of an intermediate way. So what that tells us is that the sperm, the total movement properties of a sperm population are derived from the individual behaviors of each sperm cell, but they also have a collective averaged effect. And that's important because it influences the way that sperm search space. So we also, in these simulations, quantified how much space was covered by each individual sperm. And what we found was that the pattern distributed in a manner that was very strongly correlated with their root mean squared displacement. So these progressive and intermediate sperm searched the most space, about 85% of that simulation space. And interestingly, the mixed population searched space that is very much related to its overall movement. So it's sort of in an intermediate phase, about 70% or so. And then having lower motility sperm results in them searching less space. Now, real sperm aren't just necessarily searching space for the sake of searching space. They're searching for an object. They're searching for an egg. So we also wanted to kind of explore that possibility or how that process is working. So in each of these simulations, we're increasing the total number of sperm, and all the sperm move with just a linear progressive type. So here's one sperm searching space for an egg, and here's 100, here's 1,000, and here's 10,000. And then we're simulating the egg down here in the corner by having this little pixel. Now, what happens is when the first sperm in the simulation finds the egg, the algorithm traces the path that that sperm took back to its initial position. So the takeaway here is one sperm searching in that space has to search a lot of space before it finds the egg. But when we have 100 sperm, it's still a fairly circuitous path, but it's a little bit shorter. And then when we have 1,000 sperm, it's even shorter. And when we have 10,000 sperm, then it seems to be converging on probably the shortest path to the egg. So by increasing the number of sperm, we're increasing the probability of an egg, or sorry, of a sperm finding an egg at the shortest path length. So if we plot the sperm density versus the path length to the egg, then what we can see is that there's a nonlinear drop off here. As we increase the density, then the sperm population gets nonlinearly better at finding the egg over a short path. And that above some particular cutoff, any cutoff here, you don't really have much of a benefit from having more sperm there. Now, we also thought that those sperm were searching in an open space for the egg, so their search task wasn't very difficult. But like I showed in the earlier slide, the reproductive tract is actually quite convoluted and a very complex space. So how are those relationships affected by the spatial complexity of the environment that the sperm are searching in? So in order to investigate that, we created environments that had more spatial complexity. So we chose mazes, or more specifically labyrinths, to investigate this. And the reason that we chose labyrinths is because we can quantify how complex these different environments are by counting the number of decisions that something traversing that space has to make. So for example, if a searcher starts here and it comes down here, it now has to make a sort of a decision to either go left or to go right or to go back into its initial position. So the more of these intersections and dead ends, the more complex the space. And we can quantify that by counting the node degree. So we would call this a node, and then more nodes means more complexity. So this is a plot that is the sperm agent density versus the path to the first sperm that found the egg, the path length. And we put this on a double logarithmic scale so that we could linearize that relationship rather than looking at it as sort of an exponentially decreasing pattern. And this allows us to compare these different conditions a little bit more easily. So I apologize for there not being a color scheme here, but the node degrees correspond to how they're ordered on the graph. So this is the most complex, this is the least complex. Now, what's interesting here is that the intercept is higher, meaning that the path lengths are actually longer for these different environments. But the relationship between density and the path length seems to be the same because the slope of these lines is the same in each case. And this is nice because we can actually use these relationships to generate a general equation that can describe the relationship. So the path length is equal to 10 to the intercept times x, the density, to the power of the slope. This is called a power function. Now, because they had different path lengths, it was making the relationships between them a little bit difficult to interpret. So what we did was we divided each path length by its true shortest path length, which is known because we know what's in the simulation environments. So by doing that, then we get rid of that difference in the y-intercepts, and all of these functions sort of fall into one set of lines that are more similar. And that enables us to then generate a single equation for these conditions and for these simulations that can predict the path length approximation, the ability of the sperm population to approximate the shortest path to the egg, independent of the complexity of the environment. Now, in those simulations, all the sperm behave essentially the same way. We know that in physiological systems, the sperm really don't work like that. So there's lots of variability in individual sperm phenotypes. So this graph shows intracellular calcium oscillations in distinct patterns that were observed among different groups of sperm from the same sample. So different sperm differ in their calcium oscillations, and those calcium oscillations are related to sperm behaviors through that signaling pathway that I showed you in the earlier slide. So in order to give our sperm more complex behavior, we wanted to add an oscillator function to each individual sperm agent. So for example, here are two different sperm. One has a higher frequency oscillator and one has a low frequency oscillator. And what you can, the way that you can think about these oscillations is this is proportional to intracellular calcium. So the intracellular calcium for this one starts low, and over a certain period of time, it comes up to its maximum, and then it drops back down. And it'll do this with these regular periods of time. Now, in order to couple this to their motility patterns, we created a rule that when intracellular calcium is above 95% of their maximum, then they have the opportunity to transition between motility states. And then we encode those transitions using something called a probability lookup table. So each motility state has some probability of changing to each other motility state, and then which motility state a particular sperm is in depends on what state they were in previously. And we chose the probabilities so that the behaviors, the proportions of sperm with different motility states over time were correlated with observed patterns in real sperm. So for example, they start mostly progressive linear, and then they sort of diverge into a diverse population of different motility types. But then over a long period of time, they mostly converge to being relatively low motility overall in the population and mostly weak and slow motility. So the next thing that we needed to try to figure out was how do we know how to distribute these different oscillation frequencies among the sperm to begin with? And so we developed a set of experiments to be able to quantify the distribution of intracellular calcium for large numbers of single sperm. So we used these multidimensional culture arrays to be able to sort of take a sperm sample, split that sperm sample up into different wells, and then do pseudo titrations of the signaling inputs that make them change their intracellular calcium states. So free extracellular calcium and bicarbonate, which is an important signaling effector that sperm respond to. And then we measure their intracellular calcium using different intracellular calcium dyes, such as Endo or Fura. I know this is kind of a busy graph, but this shows the averaged responses of intracellular calcium in response to the pseudotitrations over time. So if we just go to this at 2 hours of being incubated in these conditions, when no extracellular bicarbonate is there, then we don't get a very robust response to increasing extracellular calcium. Whereas when bicarbonate is there, the signaling input, then we get a pretty robust and positively correlated response to increasing extracellular calcium. Now, these effects are averaged, so there are lots of sperm within one well, and so we can't distinguish among individual sperm. So the next thing that we did was we developed some flow cytometry assays to be able to take these culture arrays to a flow cytometer and then look at the distribution among about 150,000 or so individual sperm cells with pseudotitrations of their signaling inputs like bicarbonate. So what you're seeing here is on the x-axis, increasing intracellular calcium, and on the y-axis, the approximate frequency of sperm within that subpopulation that was measured. The important thing to take away from this is that these distributions are not symmetric. So there is an average, but the average is actually skewed down here toward lower intracellular calcium. But then in each of these scenarios, there's this population of sperm cells that have relatively high intracellular calcium, which should be associated with faster transitions through different motility states. So then we wanted to kind of put this concept to work in the simulation. So now we can generate different distributions of sperm phenotypes within the initial population that's searching space. So as our control, we have a distribution free progressive. So these are just all linear progressive and they never change their motility type. But in this, this is a Poisson distributed group. And so the calcium oscillation frequencies are different for each of the sperm, and they occur within the population following this histogram. Here, the oscillation frequencies are exponentially distributed. So that means that most of the sperm have very low calcium oscillation frequencies, but there are some rare, very high calcium oscillation frequencies. And what we would expect from this population of sperm is that they should sort of devolve more quickly into a low motility pattern because they're more likely to reach motility state transitions than their low frequency oscillation compatriots. So here's those simulations. And what we found, interestingly, was that when we make that plot of the agent density versus the path length approximation, again, the double logarithmic plot, the slopes of the different lines change, which tells us that we, by changing the distribution of sperm phenotypes, we have fundamentally changed the relationship between sperm density and their ability to search as a population, which I think isn't too surprising because ultimately we've changed the frequency of sperm that are likely to find the egg, that have better search capabilities. So then we asked, well, what if we just trim some of that data off until the slopes become the same? Then that enables us to identify a critical value threshold at which the kind of lower fitness or lower search capability sperm distributions, these two relative to this one, become like the control distribution and become better searchers. So the simple takeaway from this graph is if your sperm suck, you have to make more of them. But using this sort of approach, we can figure out exactly how many more we actually need because of the relationships between the proportions within the population and the ultimate effect on the search process. So putting this together, we're very interested in developing a more formalized theory of sperm selection. So there's a theory of sperm competition, which I won't get into, but it really doesn't make solid predictions about what should happen for any particular fertilization event. It's more about the relationship over evolutionary time among competing male organisms. So to sort of recap what we showed in the previous models, let's say we start with this simple population of sperm. So there's purple sperm and there's gray sperm, and they occur in a 50-50 distribution. So the probability of seeing either trait QI is 50% or 0.5. Then let's say we put that sperm through a selective pressure, like they have to swim through a barrier, or they have to locate an egg, or they have to evade an immune system. It really could be anything. Then the population ultimately changes both in its number, its absolute number, and also its distribution of phenotypes, because there might be something about being purple that makes these sperm more likely to be successful under this given selective pressure. So in this case, all our purple sperm were retained, but only 2 gray sperm made it. So the conditional probability of observing a gray sperm, given that they were successful, is only 0.25 now, whereas the conditional probability of observing a purple sperm, given they were successful, is now 0.75. So those probabilities alone are not enough to quantify sperm fitness. And I'll show you why. What we really are interested in here is the inferential probability, the probability that a given sperm in this population would be successful given that it has a particular trait. And in order to make that sort of prediction, we need additional information. So for example, we need a prior hypothesis of how likely any individual sperm is to be successful to begin with. So we call that the prior. And then we need a likelihood. So we need to have seen some of this process play out and be able to associate the phenotypes with success. So that's actually what we're gathering down here. So by observing fertilization under selection, then we can generate the likelihoods that we need to solve this problem. And then finally, we need just a normalizing factor, basically the total probability of success, because not all the sperm in the final population are represented in the initial population. And together, this is just a simple formulation of something called Bayes' theorem. So we had done a bunch of simulations with Poisson distributed sperm in different microenvironments with different degrees of complexity. And then we looked at the cumulative posterior probability distribution. So we call this a posterior probability. So the cumulative fitness, you could sort of think about it that way. Now, in the least, notice that all 3 of these populations actually start out the same way, but they're only in different microenvironments. So the sperm population in the least complex microenvironment actually has the broadest distribution of phenotypes that can make it successful. Now, if we increase the complexity of the environment a little bit more, that makes this a little bit steeper, which means we decrease the range of phenotypes that can be successful. And if we make the environment even more complex, then we make this even more steep and we decrease the range of successful phenotypes even more. So the microenvironment acts to select sperm that have high fitness within that environment. Now, another thing that we're also interested in is being able to quantify the magnitude of selection by the environment that's acted on the change in population distributions. So I won't get into the math here because it's a little bit more extensive, but we're using this metric called Kullback-Leibler divergence, or it's also known as relative information gain. What it does is it helps us understand if we have a distribution of traits, an initial distribution like this one, and then we have a distribution that's sampled from that initial distribution that has a different shape, like this one, we can use this number to quantify the distance between these 2 distributions. And the larger that number, the more we have sort of learned about the selective effect on the population. So in our simulations, I think we ran about 200 simulations or 100 simulations, and we quantified the relative information gain with Poisson distributed sperm oscillation frequencies, and looked at the effect of increasing environmental complexity. And what we saw was that the average relative information gained increases as the complexity of the environment increases as well. So the takeaway here, or our prediction, is that you really need to know three things to understand sperm selection: the distribution of traits in the initial population, the selective effect of the microenvironment, and then how the trait distribution is related to success within that microenvironment, which we can define using Bayes' theorem. Now, if you're interested in reading a little bit more about what we did specifically with these models, we have a preprint out in bioRxiv, and I definitely encourage you to check that out. It's also in submission at another journal right now, so hopefully we'll improve that and get it published shortly. So I showed you a whole bunch of computational modeling work, but obviously those are just predictions, and we really want to be able to test this in the real world. So we've had to develop some new concepts in order to be able to do that. The first thing we focused on is making low-cost microfluidic devices, like the one shown here, that enable us to do something similar to what we were doing in the models. We want to be able to create spaces by our own design that sperm have to search, and then we want to be able to monitor how the sperm are searching within that space and do experimental manipulations to see what changes those search properties. So these devices enable us to inject sperm into an antechamber, and then they don't occupy this other space. So we can inject them in here, stop the flow, and then watch them as they sort of explore of their own accord, and then analyze the way that that works. And in order to do that kind of analysis, we actually have to develop new ways of quantifying sperm motility and pulling that data out of these complex videos, because there really isn't anything that can do that at this point. So we've been working with David Hart in computer science at ECU to develop strategies using computer vision to track sperm trajectories when there are high density cellular environments and lots of sperm crossing paths. This is a really, really fun project and has some really exciting things going on. I think that I could probably do an entire talk on just this subject, but I'll save that for another day. And in this slide, I often have a little bit of trouble getting this last piece to load, and it's currently not loading right now, so I might have to fiddle around with this. Let me go ahead and do that. The last slide is a I'm restarting PowerPoint right now, and I think this should fix the problem. I apologize for the delay. Okay, now I'm back. So we have our low-cost microfluidics devices. We have our computer vision. And then finally, we want to understand more about the distributions of physiological internal states of the cells. So we've been developing flow cytometry methods to be able to track the internal states of the cells, things like intracellular calcium, in response to controlled extracellular environmental changes, and also their evolution of those processes over time. So what you're seeing in this, and I apologize, my slide is frozen again. So I'm going to go ahead and skip that slide. I'm not really sure why it keeps freezing, but I'm waiting for the program to allow me to restart the program. It's thinking. I apologize. So the exciting thing about the flow cytometry is that it will enable us to understand not just how the phenotypes are distributed within the cell population, but also how those phenotypes change over time and how they might be distributed among subpopulations. So groups of cells that may behave similarly. Which is very important for being able to quantify the relationship between internal physiological states and the success during fertilization. So now I have closed my PowerPoint slide and I'm reopening my PowerPoint slide, and I just have one more slide to talk about. I'm so sorry for the technical difficulties. I'm not sure why it keeps freezing. Okay, so my last slide is an acknowledgement slide. I just want to thank the folks who are doing this work, as well as my collaborators. So let me switch my laser pointer. Ben Brizard is spearheading the flow cytometry project. He's a PhD student in the lab. Kylie Cashwell is also a PhD student in the lab. She's spearheading the computer vision side of the project. Ethan Caro is a master's student in the lab who started as an undergrad, and he's actually working on a different set of projects related to the complex distribution of metabolism within multicellular tissues or tissues with multiple cell types, such as the epididymis and the testis. And then a quick shout out to some really fantastic undergrads that contributed to the work as well. So Logan Harrison, who's currently applying to med schools, he worked quite a bit on the microfluidics platform as we were designing it. Chelsea Dennis is now applying to the PhD program in chemistry here at ECU, and she worked quite a bit on the microfluidics as well as some of the early agent-based modeling stuff. Stephanie Stewart also worked on the agent-based modeling project, and she's now in the PhD program in biomathematics out at NC State. And Aidan Charles worked with Ben quite a bit on the flow cytometry project, and he's now out or over at the Brody School of Medicine in their physiology PhD program. We have some fantastic collaborators. Chris Geyer in anatomy and cell biology at the Brody School of Medicine. David Hart in computer science, who's really driving our computer vision work forward. Paul Voss in Department of Public Health, who we consult with regularly about biostatistics, and who also helped us kind of figure out the information theoretic measures for selective pressure during fertilization. And we're doing a bunch of really exciting work with Martin Beer to try to better understand the implications of the sperm search from sort of first principles in statistical physics. We want to thank our generous funding from the Thomas Harriot College of Arts and Sciences, from the National Institutes of Health at NICHD, and from the Office of Naval Research. And if you're interested in any of our work, check out our lab website at caslab.com. We're on X at Cas Repro Lab, and then GitHub for any of the code for the models is available. And if you want to see videos of sperm and computer vision, check out our YouTube. And if you're in the Greenville area and you want to make $25 or know somebody that wants to make $25, we need sperm donors for our human subjects research. So thank you so much for listening. I apologize for the technical glitches. And that's the end of the talk.